Now, these homologous chromosomes in their pairs, this is two homologous pairs here. So here's a homologous pair, there's a homologous pair. The red chromosomes came from the mom, and the blue chromosomes came from the dad. So they have these pairs that form. Now, if we're starting with 4 chromosomes and we want to end up with 2, the first step would be we need to separate these homologous pairs. So they actually the pairs move, or the tetrads move to the middle of the cell. Now, how they end up here is random. For example, that little dotted line represents the middle of the cell. Now, you'll notice that this one at the bottom, I have the maternal or chromosome from the mom on this side, and the one from the dad on this side. But it very, very, very easily could have been reversed. I could have had the dad on the left and the mom on the right. Every single time a homologous pair, lines up in the middle of the cell, it's totally random which side the paternal chromosome is on and which side the maternal chromosome is on. Now, this bottom tetrad or homologous pair is totally independent of this top one. This top one, for right now, has the paternal or the dad's chromosome on this side and the mom's one on this side, but very easily, likewise, it could be reversed. However, each pair lines up along the middle is totally independent of the next one. For example, how these align is totally independent of how these align. And this is Mendel's law of independent assortment. In humans, we would have 23 pairs lining up down the middle of the cell. And because there's so many different options, it's actually 2 to the 23rd power. There'd be 8 million different combinations of how these chromosomes align along the middle of the cell. However, Mendel's law of independent assortment is huge when it comes to discussing variation or differences in offspring, which we can talk more about in class, but Mendel's law of independent assortment plays a major role in variation in offspring in a population.